Hi students! Today we're going to be talking about translation. When we had left off, we had discussed transcription, which was the process of making the mRNA transcript from the DNA. We had talked about splicing and processing, where we got that piece of mRNA ready to leave the nucleus. And now we're really going to be zeroing in on this final stage here of translation, where the mRNA transcript connects to the amino acid that we want to add to the protein. And remember, this happens at the ribosome, and it involves all three kinds of RNA. So mRNA will be there, tRNA will be there, and rRNA. They'll all be part of this translation portion. So following the process of transcription, we have created a long line of mRNA from the 5' end with the guanine cap to the poly A tail on the 3' end. And we now take that piece of mRNA out into the cytoplasm and to the ribosome. So this could be a ribosome attached to the rough ER, or it could be a free ribosome floating in the cytoplasm. Either way, the mRNA shows up and it's going to be read one codon at a time. So a codon is a group of 3 nucleotide bases. So if you look at this example here, we have the 3 nucleotide bases A, G, and U. Those are called the codon. So that is the piece of mRNA. And they are going to complement a piece of tRNA. So the tRNA is made of a whole bunch of RNA bases here. But right at this far end, at the anticodon, it has the complement UCA. So A goes with U, G goes with C, U goes with A. And this anticodon connects to a particular amino acid, in this case, serine. We're only really going to be learning the amino acids by their 3-letter name, so it connects to SER, and that's fine. The next thing that would happen is that another piece of mRNA would have another codon. So in this case, the codon is UAC, and it would have the complement AUG there. And then that would connect to the amino acid tryptophan, in this case, TYR. So what we can do is we can read from the DNA to the mRNA codons to the tRNA anticodons, and we can find the proteins as well. So we're going to just kind of be practicing that. We're going to be looking at a codon chart like this one in a minute in order to figure out which base we would match up. So for example, for serine, that's AGU. So we start with our first base here, and we would go A, and then kind of like a times table, we come across A, and we come down G until we're at AG, and then the third base is U, so we come over here. So AGU gives me serine right here, SER. So that's how we know that connection. So we're going to be practicing this for just a minute. And so what I'd like you guys to do is go ahead and write down this piece of DNA code here. And we're first going to make it into an mRNA codon. We're going to make it into a tRNA anticodon. And we're going to change this mRNA into being a group of amino acids. So we're going to change it into being a protein. So we're going to do all those things right now. So I've written down the DNA code that is on the slide. So it goes ATA, CGC, AGT. And so this is what our original piece of DNA would have looked like. And so what we're going to do now is we're going to look at what would the mRNA complement to this look like. So we know that A goes with T if we're in DNA, but we're actually making RNA, so we need to use the rule that A goes with U. So my first base of my mRNA is going to be uracil, U. T, when it's present in DNA, is still complementary to A, so I would get U, A, U for my mRNA triplet here. And then C is complementary to G, so I would get G, C, G. And then last but not least, again, A with U because it's RNA, so this would be U, G with C, T with A. So I will definitely be asking you to do this kind of formation of the complement. This is called the genetic code. This is called the mRNA codon, and that is helpful in a question. It might say, what is the mRNA codon? And then the last thing we want to look at is once this mRNA makes it to the ribosome, once this piece of mRNA makes it to the ribosome, the anticodon comes in and matches up with it. And so we could look and see what would the anticodon look like. So this is going to be our anticodon. And I just want to add a hint here that says that the anticodon is the same as the DNA code. The only difference is that it has U instead of T. So the anticodon is going to be the same. So again, we do our base pair match here. So we're going to go with our U. U goes with A. A goes with U. U goes with A. G goes with C. C with G. G with C. U with A. C with G. A with U. So what we end up with, our TNA anticodon, is almost the same as our DNA, except we have substituted in U's where before we had T's. So that's a fast way. If they give you the DNA to get to the tRNA, you can get there a little faster. On the test, like I mentioned, I may give you the DNA and ask you for the mRNA. I may give you the mRNA and ask you for the tRNA. I might give you the DNA and ask you for the mRNA. Those are all things you should be able to do so far. When you're doing this, it's really a factor of keeping in mind just these base pairing rules and making sure you know whether you're making a piece of RNA or DNA. So what we're going to do now is we're going to look back at that same piece of mRNA that we transcribed just a little bit ago, and we're going to translate it into a protein using this chart. So I just want to go through the chart again. Our first position in our code is here on the left. So if I were starting with C, for example, I would take this C and I would go across the chart until I ran into whatever my second base was. So if my codon I was looking at was C, 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 I would start here with my C's and I would come across till I got to my next column, which had a C, and then finally my last column, which had a C, and I would be right here at PRO or proline. Okay, so you should have an mRNA list in front of you. It should be from our previous slide, UAU, GCG, UCA. So I want you to find that piece of mRNA and I want you to rewrite it somewhere down lower on your paper so that we can practice translating. Codon that we're looking at is UAU. So what we want to do is we want to go to where U is, where A is, and where U is. So TYR is my first amino acid that I'm making. So this is my amino acid. So my first one is TYR. My second amino acid that I would make goes with G, C, G. So we go G down here, over to C, and then down to G, and we get ALA. And last but not least, we're going to go U, C, A. So my third position is SER. You're going to notice a couple of special codons here. UAA, UAG, and UGA all mean stop. So when the protein gets a code like UAA, there is nothing here. And so that means that we're done. The protein's finished. And so there's no link. That's how we release the protein for it to go. Also, when making a polypeptide, we are going to start always with AUG or methionine. So methionine is the start codon AUG, but it codes for the base methionine. These amino acids get added link by link by link in order to form a polypeptide. Remember that the bond that forms between the different amino acids is called a peptide bond. So we call this a polypeptide because we're forming many peptide bonds. And this polypeptide then eventually gets folded and helped and formed into a protein. So we're forming the very basis of that polypeptide chain. So each tRNA needs to both break a bond and form a bond in order to keep the polypeptide growing. So we're going to look over here at this growing polypeptide chain. So at the moment, we have 1, 2, 3 amino acids in our polypeptide chain. What we're going to see happen next is a new tRNA is going to come in and its anticodon is going to match the codon here. And we're going to form a new bond, a new peptide bond between the third and the fourth amino acid. And at the same time, we are going to break this bond that is between the amino acid of 3 and the third tRNA. So now the tRNA is free and it can leave, and my growing polypeptide is now attached to site 4. At that point, the ribosome scoots down. It shoves down, and now a new tRNA, the 5th one, can come in and sit next to the 4th one, and we can get the same thing over. The connection of a polypeptide bond between 4 and 5, and the breaking of the bond between that amino acid and the tRNA. And now we get our 5th amino acid. And so our polypeptide is growing as each amino acid gets added. Eventually, as you know, we're going to hit a stop codon. We're going to hit that UAA codon. And when that happens, the tRNA that comes in does not have an amino acid attached to it. And so instead of forming a polypeptide bond, it does not form a polypeptide bond. It just simply releases the polypeptide to go be formed and transitioned into a protein. The rRNA is behind this, catalyzing it. So we've been focusing on just the mRNA here in the dark blue and the tRNAs, which in this case look like little forks. But behind them is this big rRNA, or the ribosome, which is giving the energy and forming, allowing for the formation of that peptide bond and the breaking of the bond between the amino acid and its tRNA. So we're now to the point of kind of reviewing what we have been going over. I want you to take a second and just kind of look at this diagram and start to see all of the pieces that you have learned that are happening here. Let's focus on the DNA first. We see RNA polymerase here, and RNA polymerase is catalyzing the formation of the blue mRNA, and it's doing that by using free nucleotides that are in the nucleus. It adds them together, and eventually our mRNA is going to be spliced and processed, and it's going to leave the nucleus and travel out to the green rRNAs here, and you can see the formation. Remember that the code for these ribosomes is also held in the DNA, so the rRNA and the proteins link together into this ribosome. And our tRNAs are also here with their anticodons, and they're connecting back here to amino acids. Those amino acids come from the food that you eat. You know that you need to eat protein to build protein. This is why these amino acids get broken down into these little pink squares in your digestive system, pulled in through your blood, given to your cells, and are now attached to tRNAs so that they can be added to this elongating polypeptide chain. We have the tRNA coming in. Our next tRNA comes in, we move the amino acid polypeptide chain over, and eventually release that polypeptide chain to go be a protein and do something important inside of your cell. So we have successfully gone through the mechanisms and the controls over the mechanisms for gene expression. How do we make the protein and when do we express the gene? You guys have really covered a lot of complex information. The last thing we want to look at today is mutations and how those mutations affect this protein product. Mutations in the DNA, in the mRNA, how they eventually can impact the proteins that you are making. Any change to your DNA is considered a mutation. We are predisposed from X-Men and other movies to think of a mutant as a bad thing, but we are all mutants. We are just a series of mutations that have led life from unicellular prokaryotic life into the complex eukaryotes that we are now. We are mutants. We all are. Mutations can be good or bad depending on the environment that they're in. For example, if you gained a mutation that allowed you to breathe underwater, that would be a good mutation if you lived underwater, but it would be a very bad mutation in the desert. So it's not the mutation that is good or bad, it's its usefulness in your environment. So all mutations come from one of these kind of mutations here. We can see a substitution mutation. If we substitute in the wrong nucleotide, so what we're picturing here is that this is a piece of mRNA in green, and it is being translated 3 nucleotides or 1 codon at a time into amino acids, and the amino acids are being represented by the shapes. So what we see here is in our 3rd codon, it used to be UGC, and that was coding for this orange triangle. And now it is UGA, and unfortunately that provides a stop. So that is a single nucleotide base substitution that ends up causing a really big effect. We were going to make a long protein, but instead now we've stopped. They call that a truncated protein. So even a single base pair mutation can cause a huge ripple effect in your proteins that you make. We can also see deletions or insertions, and what those cause is actually a frame shift mutation. So you can imagine you've taken out or added in a base, and so every single codon from then on is going to be messed up. So we can sometimes see mutations that are silent. In the genetic code, there is plenty of redundancy. So if we had a mutation, a substitution that changed CCU to CCC or CCA, that would be a silent mutation because you were going to make proline and now you're still making proline. So it doesn't really make any difference. You can have a mutation that causes just a single nucleotide change. So maybe you were going to be CCU, but now you're CUU, so where you were going to be proline, now you're leucine, and so that can have a various amount of effect on the cell. Most importantly, though, is when we accidentally get those stop codons and we truncate the protein, or when we insert or delete one of our bases, and so we've caused a whole frame shift, and so every amino acid from then on is going to be wrong. That's when we start to see usually bigger errors in our cells. So at this point, we've really finished up most of eukaryotic gene expression and control. But we do need to take a moment and talk about prokaryotes, and we're going to do this when we meet up again. Good job today, you guys, and I will see you in class.